Hello everyone. Children, today we are going to start with a new chapter, that is Structure and Germination of Seeds. In the previous chapter, you have studied about the pollination and fertilization. You have studied about the different types of pollination, the agents of pollination, and how the anther, or we can call that pollen grains, reaches the stigma. Fertilization occurs, and then after fertilization a zygote is formed. In that process, the ovary changes into a fruit and ovule changes into seed, or we can call that it as a mature ovary is called the fruit, and mature ovule forms the seed, or it is known as the seed. So here children, in flowering plants the seeds play a very important role. So now we will study about the seeds. In life of flowering plants, seeds are very important structures as they help in the formation of new plants. See children, we all know that seeds are very important and they form the new plants. Without seeds, it is difficult to form new plants. So, after fertilization, we get the fruits and seeds. A seed is a mature ovule after fertilization. So, after fertilization, the ovule becomes mature and is known as a seed. The ovule develops rapidly after fertilization using the food reserves. After fertilization, this ovule, it grows rapidly by utilizing the reserve foods present in the endosperm. In addition to the development of new plants, seeds help in the dispersal of species as well as overcoming unfavorable conditions. So children, these are the functions of seed. We have seen these functions in the previous chapter also. So seed plays an important role. First thing, it helps in the dispersal. Secondly, it protects the baby plant which is present inside the seed from unfavorable conditions. And these seeds also forms the food. It also contains the food inside it. Now we will study about the structure of seed. All seeds have common features, though they vary in size, shape, color, and some other features. A typical seed is differentiated into three parts. See, children, seeds are of different shapes, different sizes, and different colors. But all the seeds, whether they are different in their structure, in their shape, in their size, in their color, but they have some common features. Now we will see what are these features. A typical seed contains a seed coat, embryo, and stored food. So the seed is differentiated into three parts, that is the seed coat, embryo, and stored food. So these are the three parts of a seed. Now we'll see the seed coat. Each seed is covered by a protective covering called the seed coat. It develops from the integument of ovules. See children, when we were studying the structure of ovule, we have seen that ovule is surrounded by two layers. And these two layers are known as the integuments. So, these integuments, they forms the seed coat. The seed coat is differentiated into an outer tough protective testa and inner thin tegmen. See, children, the ovule is surrounded by a double layer. It is well protected by a double layer, and that double layer is known as the integument. And these integument, it forms the seed coat. Seed coat means it forms a protective covering over the seed. So the function of the seed coat is to protect the seed. The outer integument, it forms the is tough and it is called the testa. Inner is thin and is known as the tegmen. So the outer integument is tough, it is thick and is known as the testa. The inner integument is thin and is known as tegmen. And both this testa and tegmen together is known as a seed coat. The testa protects the seed from desiccation, bacteria, fungi, and insects. So, this seed, the function of this seed coat, or we can call it the function of the testa and tegmen, is to protect the seed from bacterial infections and insects. On the seed is present a scar called hilum. So, the seed has one scar. That scar is known as the hilum and it represents the point of attachment of the seed with its stalk. So this hilum, it represents the attachment of the seed with its stalk. When the seed is attached with its stalk, that is known as that it forms a scar, and that scar is known as the hilum. Adjacent to the hilum is present a minute opening called the micropyle. Adjacent to the hilum, adjacent means lying next to the hilum or very close to the hilum, is a minute opening, and this opening is known as the micropyle. See here, children, this is the structure of a seed we can see here. These are the testa and tegmen. And this one, a scar you can see here. This is the scar. And this scar is known as the hilum. And lying next to the hilum or adjacent to the hilum is a small pore, and this pore is known as the micro. You can see here, children, this is known as the micropyle. Understood, children? This is the point of attachment. It is attached with this stalk with this at this place, and this is known as hilum. And here you can see a small pore. It is not visible, but here it is a small pore, and this pore is known as the micropyle. So this is the structure of a seed. Now, so function of the hilum is, it is the point of attachment of seed with its stalk. It is attached to the seed with its stalk at this point. Then micropyle, absorption of water as well as exchange of gases take place through the micropyle during germination of seed. See this through this micropyle. Micropyle is a small opening which is lying adjacent to the hilum. And the function of this micropyle, first function is absorption of water. Through this opening, the seed absorbs the water when it is soaked in water. And second function is the exchange of respiratory gases takes place through this micropyle during the germination of seed. When the seed starts germinating, what happens, children? This exchange of the respiratory gases takes place through this micropyle. So this micropyle has two functions. First one, it absorbs the water, and secondly, exchange of respiratory gases takes place through this point. The micropyle is not easily seen in a dry seed. See, this micropyle you cannot see easily in a dry seed, but if the seed soaked in water is pressed gently, some water and air bubbles ooze out through this pore and it can be easily located. So children, if you want to see this micropyle, you soak a seed in water, and then after soaking it overnight, you gently press that seed. So you can see from the micropyle, the water will ooze out and you can see some air bubbles also, and then you will understand that this is the micropyle. So you can soak the seed in water and you can see. Next is, children, the embryo. An embryo is defined as a young miniature plant along with cotyledons enclosed within the seed coat. So inside the seed is a baby plant, and that baby plant is known as a embryo. It is known as embryo, and this embryo is enclosed along with the cotyledon. See, embryo is defined as young miniature plant. What is embryo? It is a young miniature plant along with cotyledons. See here, these are the cotyledons. Along with the cotyledons enclosed within the seed coat. So the cotyledons with the miniature plant enclosed by a seed coat is known as a forms the embryo or is known as a embryo. The embryo is differentiated into, now this embryo is differentiated into a cotyledon and embryo axis. Cotyledons and embryo axis. Now see here children, cotyledons, also called the seed leaves. See here in this is the structure of a generalized seed, and this seed is cut open. See, these two are known as the cotyledons. They are also known as the seed leaves. See here, you can soak the bean seed in water and then you can remove the seed coat. And when you will remove the seed coat, you can open that two cotyledons on either side, and inside in between them you can see this embryo axis. This is the embryo axis. So we are studying about now cotyledons. So these cotyledons, they are also known as the seed leaves. They are either two in dicotyledonous plants or one in monocotyledonous plants in number. So if a seed is having two cotyledons, it is known as a dicotyledonous plant. And if it is having only one cotyledon, it is known as monocotyledonous plants. They are food-laden, fleshy structures attached to the embryo axis. So these cotyledons, they are food-laden. That food-laden means they stores the food. And they are fleshy structure, and to these cotyledons an embryo axis is attached. You can see this is the embryo axis. That means these cotyledons are attached to this embryo axis. They provide nourishment to the embryo in the early stages of development. So the function of cotyledon is to provide nourishment to this growing, we can see, to this embryo in the early stages of its development. When the germination takes place, what happens? These cotyledons, they provide nourishment to this growing embryo. In endospermic seeds, cotyledons are not massive. Endospermic seeds, see, some are endospermic seeds and some are non-endospermic seeds. So in endospermic seeds, these cotyledons, they are very thin. They are not massive. They are very thin, and the nourishment is provided by the endosperm to the embryo. So in endospermic seeds means the seeds which have endosperm, and this endosperm provides nourishment to the growing embryo. In endospermic seeds, cotyledons are not massive. The example is castor. So in endospermic seeds, cotyledons are not massive. The food is stored in the endosperm. Now about the embryo axis. It is the young or miniature plant differentiated into following parts. Now you have seen this is a generalized seed here. You have seen these two cotyledons. They are attached with the embryo axis. So now we will study about the embryo axis is differentiated into following parts. First one is the mesocotyle. It is a part of the embryo axis where cotyledons are attached. See here, this portion, these are the two cotyledons. They are attached to each other with this axis. And this stalk, we can call it as the stalk, and this stalk is known as the mesocotyle or the cotyledon stalk. So the function of the mesocotyle is to attach the two cotyledons. Epicotyle, it is a portion of the embryo axis between the mesocotyle and plumule. See here, you can see this, children. This is the plumule, this one. This is the plumule and this is the mesocotyle. So the portion between the plumule and the mesocotyle is known as the epicotyle. This is the mesocotyle, this bridge-like structure. This is the mesocotyle and here this is the plumule. So the portion between the plumule and the mesocotyle is known as epicotyle. Hypocotyle, it is a portion of embryo between mesocotyle and radicle. See here, the portion between the mesocotyle and plumule is known as epicotyle. In the same way, the portion between the mesocotyle and the radicle, it is known as the hypocotyle. Children, this plumule forms the shoot system and this radicle forms the root system. So here it is very clearly shown here in this figure. These are the two cotyledons. They are attached by a stalk or by, we can call it as a bridge-like structure. This is known as the mesocotyle, and the portion between the mesocotyle and the plumule is known as the epicotyle, and the portion between the mesocotyle and the radicle is known as the hypocotyle. Now the plumule. It is the feathery or leafy end of the embryo axis that grows into shoot system of the plant. See here, this is the plumule. It is here at the tip of this embryo axis and it is feathery, and or we can call it as the leafy end of the embryon axis. This is the plumule. Now about the radicle. It is pointed lower end of the embryo axis that develops into the root system of a plant. See here, radicle, you see, this is the pointed part of this. This is known as the radicle, and in the later stages it forms the root system. So these are the parts of the embryo axis. So the parts of the embryo axis are mesocotyle, epicotyle, hypocotyle and plumule. Now we will see here the food storage in the seeds. As I told you that some seeds are endospermic and some are non-endospermic. Those seeds are endospermic seeds. The food is stored in the endosperms, and in some they are non-endospermic. In that type of seeds, the food is stored in the cotyledons. Food storage in the seeds. The seed contain stored food materials to be used during the early development. Now, the seeds, they contain the stored food materials which is used during the development, or we can see during the early development. During the early development of the seed, this food is used. The food is stored either in the cotyledons or in a special food storage tissue called the endosperm. So this food is stored either in the cotyledons. If it is stored in the cotyledons are thick and fleshy. But if the food is stored in the endosperm, in that case the cotyledon is thin. The cotyledons are thin and membranous. They are not thick and fleshy. The seeds in which food is stored in the cotyledons and contain no endosperm are called non-endospermic or X-albuminous seeds. So, the seeds in which the food is stored in the cotyledons, they are known as the non-endospermic seeds, or they are also known as X-albuminous seeds. Example is bean, gram, pea. See, in bean and gram, you all have seen beans and gram. They have thick cotyledons That means the food is stored in the cotyledons. Castor, cotton, maize, wheat, rice, whereas those seeds which contain stored food in endosperms are called endospermic or albuminous seeds. So, in seeds which contain the stored food in the endosperms, they are known as the endospermic seeds or albuminous seeds. So in this endospermic seeds, the cotyledons are very thin and membranous. Now, types of seed. Depending upon the number of cotyledons and the presence or absence of endosperm, seeds are classified as shown in this table. So, depending upon the number of cotyledons and the presence or absence of the endosperm, the seeds are classified into C, depending on the number of cotyledons. If the seed has two cotyledons, it is known as dicotyledonous. And if the seed has one cotyledon, it is known as monocotyledonous. So in dicotyledonous, the dicotyledonous also have exalbuminous seeds and albuminous seeds. Exalbuminous means seeds without endosperms. They are known as exalbuminous. And examples are bean, gram, pea. Albuminous means having endosperm, castor, cotton, etc. In the same way, the monocotyledons, that means the seed is having only one cotyledon. In that, the seeds are exalbuminous. Exalbuminous means without endosperm. Without endosperm means the seeds have cotyledons. Example is alisma. Albuminous having endosperm. So the seeds, those who have endosperms, they are known as endospermic seeds, or they are also known as albuminous seeds. Example is maize, wheat, rice, etc. So children, here we end this session. This is the A part of the structure and germination of seed. Thank you.